Hi, welcome to a Physionic detailed study analysis. This is going to be a heavy one. We're going to be going into all kinds of different factors, different areas to investigate the relationship between your body fat and your muscle tissue as it specifically relates to a condition known as sarcopenia. But this is going to be relatable if you're interested in just learning about that relationship, regardless of if you have sarcopenia or not, which I'll define in just a little bit. If you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. I hold my master's in exercise physiology, and I do this. I read studies on a non-stop basis because this is what excites me. So if you're interested in learning about sarcopenia, muscle, fat relationship, how they communicate with one another, then you're in the right place, because we're going to be going over a scientific review of many studies that have been pulled together to create an overall explanation of the scientific literature. Without further ado, let's go ahead and jump right into it. So to start out, to give you an overview of what we're going to be going over, we're going to go over what is sarcopenia. Then we're going to look at the relationship between body fat and muscle. And then we're going to go into some of the mechanisms which are going to be related to inflammation. And then we'll go into how fat releases particular hormones and how those hormones affect our muscle size as well as function. And we're also going to go into a few of the muscle hormones that are also secreted from the musculature. Although this is going to be more focused on the one relationship, so fat on muscle. And then if you're part of my Physionic Insiders, you'll get access to this full analysis, which also goes over a few more hormones. And some of these hormones you've probably never heard of in your life. They're very obscure. So I'm really interested to cover those. And then I think this may be one of the first times that I've actually covered this, but going over long non-coding RNAs and microRNAs. If you're not familiar with that and you're part of the insiders, then you'll learn all about them in this episode. If you're interested in accessing the full study analysis, then click on the link and join the Physionic Insiders, where you'll get access to this and many other benefits as well. But if you're just interested in the first four topics, if you've been with Physionic for a while, you know the deal. Let's go ahead and get into it. So this research comes from this scientific review, as I mentioned, which is called Recent Advances in Future Avenues in Understanding the Role of Adipose Tissue or Fat Tissue, Crosstalk in Mediating Skeletal Muscle Mass and Function with Aging. So where does that aging component come in? So that is actually sarcopenia. So when we discuss sarcopenia, we're really just talking about the age-related loss in muscle size. So it can be defined by the actual muscle size, which is what most people attribute, but also to the function. So can you walk? Can you get up from a seated position without the assistance of your arms, from a sit to stand test, things of that nature that actually identify the function of the muscle, which, as I've argued in the past, is probably, and I still hold this view up to now, is that function is probably more important than the actual size of the musculature, although they're tightly related to one another. Now, what's intriguing is that, and something that most people don't consider, is the fact that your fat cells, the adipocytes, can actually have dramatic effects on your musculature, and vice versa as well. So your musculature can have positive or negative effects on your fat cells, because both tissues release certain factors. If that's cytokines, if that's hormones, whatever it might be, can be released into the bloodstream, or if they're right in close proximity with one another, can interact in what's known as an autocrine or a paracrine form of action, which is in that hormone category. So there's a lot of crosstalk between the two, and that's what we're going to be investigating. Now, with obesity, what people identify is the fact that with muscle tissue, there's actually an increase in muscle size. So the musculature itself will increase in muscle size. And that, of course, correlates with increased size of the fat cells, because obviously if you're overweight or obese, then you have higher levels of body fat. So you need an area to store all of that body fat. However, the drawback, so that alone, I mean, the fact that you see these increases in the size of the musculature, you would think, well, that's not sarcopenic. That's exactly the opposite. So it's, I mean, I don't want to say a good thing to be obese, but there's a benefit to being more overweight. But that actually turns out not to be the case. And this is where we start getting into the nuances of actually looking at muscle function. Because when looking at actual muscle function, obese individuals tend to have far inferior muscle function, even though the muscle size is increased. And that could be for a variety of different reasons. If that's lower protein content, contractile units within the actual muscle cells. So the actual muscle cells have certain proteins that serve particular functions. If you have maybe the muscle cell itself may be engorged, but the proteins within it may be diminished. You can have fibrosis that occurs between the muscle cells. There's all kinds of different reasons as to why the function of the musculature in totality, so this total muscle belly, would be impaired. And here we're looking at a cross section. So this would be a healthy musculature. So each one of the kind of these circles is a single muscle cell. And we can see here that there's considerable fibrosis, or you get the penetration of a series of fibrotic tissue that replaces the musculature, as well as just gets in the way of the function of the musculature. And this is just one example of something that could go wrong. And this occurs heavily with obesity. So do fat cells have an effect on this? Do they increase this level of lack of function? And in truth, the answer is yes. So not only do people experience sarcopenia from aging, but they also experience increased sarcopenia from being overweight. So it's like a double hammer. I was going to say double-edged sword, but that's not accurate. It's a double hammer on one factor, looking at musculature. So not only is aging leading to this decline in muscle size and function, but also being overweight, having overfatness, also rapidly accelerates that sarcopenic problem, which of course is a huge issue once you get into your 60s, 70s, and 80s and beyond. So what is causing this effect? What is actually causing this increased sarcopenic effect? Well, as I mentioned, there are a number of different factors, and these are just a few of them. We're going to be going over more of them, but you have like tumor necrosis factor, or you might have leptin, which you may be familiar if you've looked at any hormones related to adipocytes, fat cells. Resistin, we're not going to be focusing on resistin, but we are going to be talking about these other two as well as many others. So there is a crosstalk in terms of the release of these cytokines, if it's TNF, or hormones, if it's resistin or leptin, that end up binding and affecting the musculature, and we're going to see exactly how. But beyond that, something that people don't consider, and something I'm going to be covering later, if you have access to the full version of this, is to also look at long non-coding RNAs, which are essentially sections or particular things that we don't really actually consider when considering kind of common biology. We think of RNA, we think of proteins, we think of DNA, but a huge majority of our DNA actually encodes for or holds the information for RNA that is not going to be translated into a protein. So in one of those examples is these long non-coding RNAs. And they have a series of functions that, again, I'll go over later, that affect how our cells react to particular stimuli. I realize that's a little bit vague, but once we get into those specifics, you'll see exactly what I mean. But what's really, really cool is the fact that your fat cells can release long non-coding RNA. They can package them up into vesicles, which are known as extracellular vesicles, and kick them out of the cell. So then they're protected inside this vesicle, and it will float through the bloodstream and end up at the musculature, which will then be taken up, and those long non-coding RNAs can then have an effect by either, as one example, reducing the expression of particular genes or increasing the expression of particular genes, or having an effect on the RNA there as well. So which is a really, really cool mechanism. But this is another way, aside from cytokines, aside from hormones, that you can have this crosstalk between these two tissues. However, there's also such a thing as senescence. So your fat cells can also become senescent. So if you have white hair, for example, that is an example of senescence. So you have these hairs that have kind of gone into a dormant state. It's not completely accurate to say it's a dormant state, because these cells do still do things. They still secrete cytokines. They still secrete oxidative stress. They do a series of different things. It's just that they're not quite as active as when they are actually part of the normal physiological system. So what do we see here? Well, we see that senescent adipocytes, or fat cells, can lead to the release of more reactive oxygen species, more interleukins, which are typically, not always, and I'll show you some examples in the next slide, but are typically known as pro-inflammatory. There are some anti-inflammatory as well, and as well as the release of a series of other proteins, some of them which have been identified, some of which have not been identified, or other cytokines that have an effect on the muscle cell. And now, of course, with one senescent cell, that increases the probability that nearby cells, if they're fat cells or if they're muscle cells, if they're constantly bombarded by these added stressors from the external environment of another senescent cell, then a fully functioning cell can turn senescent, and probably will turn senescent more quickly because it's around other senescent cells. So it has wide-reaching effects, not only from the release into the bloodstream, which not only releases these molecules into for widespread effects in musculature in other parts of the body, but also in a really local fashion as well. And these are all called SASPs, which are senescent associated secretory phenotype. So if you have more of this generation of this SASP, or these molecules, then you have what's known as SASP. So we're really getting into this inflammatory area now. So looking more at inflammation and looking at some of these pro-inflammatory molecules and anti-inflammatory molecules, if you look at obesity and you look at being over fat, the way that that can have an effect on the musculature as well is by the overproduction, as a result of over-fatness, of pro-inflammatory molecules. So you're looking at cytokines like interleukin 6, TNF alpha, tumor necrosis factor, interferon, depending on the interferon that you're looking at, interleukin 1 beta, interleukin 15. Now this one we're going to go into a little bit more detail, because this is also increased. So interleukin 15 is increased, but it may not necessarily be pro-inflammatory, and we're going to go into that in just a second. However, so you see this massive spike in these pro-inflammatory molecules which activate and recruit immune cells to the musculature, which then has this compounding effect because they start releasing more of these pro-inflammatory molecules. Well, that eventually leads to an invasion into the musculature, and then you have more fibrosis, you have less muscle function, you have a series of different things which are a whole nother deep dive to go into to figure out what happens once the immune cells actually are recruited to that area. And on top of that, you start to see dramatic reduction in the production of anti-inflammatory cytokines. So the balance between the two are focused highly on pro-inflammatory cytokines. So anti-inflammatory like interleukin 10 and other interferon molecules. So interleukin 15 probably isn't a pro-inflammatory molecule in this situation, but it does increase with sarcopenia. What's intriguing, however, is with obesity and sarcopenia, you see a reduction in interleukin 15. So what's going on here? Well, when we actually look at the effects of what interleukin 15 does once it binds to the musculature, we understand why it might be increased in sarcopenia and be decreased when you have sarcopenia plus obesity. So interleukin 15, once it binds to the musculature, leads to a reduction in proteolysis, which is the degradation of proteins within the musculature. So it preserves the musculature by being anti-atrophic. So it's anti-atrophy. So it's anti-shrinking of the musculature, which is obviously one of the hallmarks of sarcopenia. And the way it does that is it actually reduces the activation or the activity in general of what are known as ligases, which are particular functional proteins that go around to non-functional proteins and tag them for degradation to eliminate them. Or they can also tag functional proteins that have been damaged or no longer serve a function or whatever it might be. And that is a vital part of the health of the cell. But if you have too much of it, then you start to have the shrinking of the muscle cells. So interleukin 15 inhibits this action. But on top of that, it also inhibits the pro-inflammatory molecules. So the thought is in sarcopenia, and this is just a hypothesis by these researchers, is that you see increases in interleukin 15 to combat the negative effects of so many pro-inflammatory molecules that are also released. Looking at interleukin 6, TNF alpha, some of the interferons that we were looking at earlier. So while we see increases in those, we also see increases in IL-15. And it's not because it's also pro-inflammatory, but it's to try to curb some of the negative effects of having too much inflammation occurring within the body. Now, one of the problems, however, is that with interleukin 15, you see really high levels of interleukin 15. I think it's something like double what you would see in healthy, normal individuals. However, sarcopenic muscle does not express much of the interleukin 15 receptor. So while you have a ton of it in the plasma, you may have plenty of it in the bloodstream, you may have plenty of it in the extracellular fluid, but unfortunately you need to have enough receptors to actually be able to be sensitive to IL-15. So in sarcopenia, you have this situation where high levels in the bloodstream, but they can't actually do anything because the receptors aren't there. You don't have enough of the receptors to actually cause the intracellular signaling within here because it needs to bind, and then that leads to a change in the receptor, and then you get a bunch of signaling within the cells, like what I mentioned earlier with its blunting effects on the pro-inflammatory cytokines is one example. Now, I did mention that in obesity, you have lower levels of interleukin 15. So there's a difference in the negativity of the mechanism. So with sarcopenia, you have high levels of interleukin 15, sure, but you don't have enough of the receptor. With obesity, I don't know they don't mention anything about the actual receptor number, but it doesn't really matter because you could have all the receptors in the world, but you don't have enough interleukin 15. So regardless, it leads to the same effect, that interleukin 15 does not have the effect that it should have, which would be, again, to curb those pro-inflammatory molecules. So there's a lot going on here in terms of inflammation. This is just a small subset of the number of things that are going on. We could certainly go into deep dives on each one of these molecules and their effects on the musculature. But I wanted to shift focus on hormones. So we're going to be looking at leptin, lipocalin, myostatin, and adiponectin. So the main fat-related hormone is adiponectin, and of course significant amounts of leptin are also produced, and you get some production of this lipocalin and some of myostatin, although myostatin is massively reduced by comparison to the production in the musculature. The musculature produces significantly more myostatin and also produces sizable amounts of leptin and does also produce lipocalin. So lipocalin 2 specifically. Okay, so to dive deeper into this, let's look at adiponectin first. So adiponectin does have an effect. So it's secreted by the fat cells, by the fat tissue. And once it does that, it ends up in the bloodstream and then has direct effects on the musculature. Well, how does it do that and what does it actually do? Well, it does that by binding to its receptor, which is found on the muscle. So this is a muscle cell. We've got our cell membrane. This is outside the muscle cell. This is inside the muscle cell. And adiponectin will then bind to the receptor on the outside, which will lead to a bunch of changes to the actual receptor, which will then lead to the recruitment of this protein right here, known as APPL. So APPL has a really positive role within the cell in that it recruits a series of other proteins. Now, these proteins are heavily involved in glucose metabolism, glucose homeostasis, as well as improved fat metabolism and fat breakdown, fat utilization, fat uptake, things of that nature, fat homeostasis. So why does that matter? Well, the reason why that matters is because these proteins are related to insulin sensitivity. So we've got proteins like IRS, we've got proteins like AKT, we've got proteins like AMPK. All of these proteins are in some way influenced by APPL. And the reason why I say that they're drawn to it or that they come to it when it's activated, when it's in its prime position, is that APPL can also be found near the insulin receptor, can also be found at the adipocyte or adiponectin receptor. And the reason why that's powerful is because it's known as a scaffolding protein. So when we think of like enzymes, they'll act on a particular molecule and then turn it into another molecule. Well, you have other proteins within the cells that serve other functions, and one of them is a scaffolding protein, meaning that it's present so that it can hold on to a bunch of other proteins and keep them in very close proximity with one another. And because they're in such close proximity with one another, there's a higher likelihood that they're going to interact as a result. And that's what APPL does. So ultimately, it takes insulin sensitivity proteins, enzymes, proteins, same thing. It takes fat metabolism proteins, it takes signaling proteins within the cells, and it holds them very close together so that they're constantly interacting with one another. And that ultimately leads to improvement in glucose metabolism and fat metabolism. So adiponectin has this effect. And of course, with sarcopenia, you see reduced levels of adiponectin. So even though, and with obesity, although you think, okay, well, I have so much more of this of fatness around me, so I have so many more fat cells, why wouldn't I be releasing tons more adiponectin? Well, it seems to be that there's a threshold, so you can't keep producing more and more and more adiponectin, which will be a slightly different story when we're talking about leptin. Another one is to look at myostatin. So myostatin is one that was all the rage for a while because they thought there were these bulls and these dogs and even some humans where there were these myostatin deficiencies. So myostatin, the way it works is that it's released. So a little bit is released from the fat cells, but primarily by the muscle cells. And it can feed back onto the muscle cells, which is known as an autocrine reaction. It can also have a paracrine reaction where it affects muscle cells a little bit downstream, or it can still act as a hormone and you can have muscle cells in your legs that secrete it, and then it affects muscle cells in your arms. So there's a number of different ways that it can function, but ultimately it does bind to muscle cells. And when it binds to the myostatin receptor, again leads to a change in the receptor, and then it inhibits protein synthesis. So you get less formation of proteins and you get more of what's known as the proteasome activation, which actually goes back to when we were talking about degradation and proteolysis. So you have greater levels of degradation and you have lower levels of protein synthesis. So that ultimately leads to a net effect of the muscle shrinking, because you're degrading more of the proteins, more of the parts of the cell than you're creating, which ultimately leads to a shrinking of the musculature. So with the deficiencies in myostatin, then you tend to see, not even 10, you really see these dramatic changes in musculature to the point where you see huge increases in muscle size, because you no longer have this break on protein synthesis, and you no longer have this stimulation of protein degradation. This is a really, really powerful hormone. Now, what's intriguing is that with obesity, you see increases in myostatin level. So the muscle cells are producing more of this myostatin, which would then inhibit the size of the musculature. But of course, there are many different mechanisms that are at play, because you can have particular growth signals that are highly elevated, and myostatin can also be elevated. So it really depends on the context, because of course, if you think about it, the increase in myostatin is completely antithetic to the idea that you see increases in the muscle size with obesity that I mentioned at the very beginning. However, it could also have an effect on muscle function. And of course, aging can also contribute to doubling myostatin levels in the body. So how do you combat this? Well, you combat it by something as simple as exercise. So exercise reduces myostatin levels. And if you obviously continue to exercise, then you're going to continuously be knocking down myostatin levels. Now, another protein that people are often familiar with when they're looking at myostatin is to look at follistatin. Follistatin is a protein that actually inhibits myostatin. So you have a series of different things that are constantly modulating, constantly changing within the body, but you're looking for overall trends. So while we see increases in myostatin, maybe we also see increases in follistatin, or maybe we see decreases. They didn't actually discuss that too much in this review, unfortunately. They only focused on myostatin. Okay, so to discuss this in far more succinct terms, now, of course, if you actually want details on all this stuff, you're going to have to go back and look at the rest of it. But fat cells crosstalk with muscle cells by releasing cytokines and hormones, and even those long non-coding RNAs, which I'll go into later in the full version, that bind to the muscle cell and change its size, function, as well as metabolism, as we saw with adiponectin as one example, and more, certainly more than what's just discussed here. Now, with obesity and sarcopenia, fat cells release far more of these pro-inflammatory molecules and oxidative stress, causing greater muscle loss. So it's one thing to be sarcopenic, but then on top of that, to also be obese, on top of that, leads to a compounding effect, which can be extremely problematic. Now, if you're interested in covering two more of the hormones, so lipocalin 2 and leptin, and how those have an effect on musculature, as well as far more information on the non-coding RNAs and the long non-coding RNAs and microRNAs, and how those affect the musculature, then just sign up for the Physionic Insiders and you'll get access to this, as well as all my other study analyses and future study analyses and seminars and office hours and all kinds of good stuff. So, and a private community, which is really fantastic as well. Anyway, thanks for stopping by. Hopefully you learned something, and I'll catch you in the next one. Have a good one. Bye.